we made these nanoemulsions. People will call them liposomes, but it's more technically a nanoemulsion of hemp oils. And we said, oh, wow, these things are really good. They really relax you. And some of the early data that we looked at, it was CBD as an anxiolytic. And so I was like, okay, it's good for that. And people talked about it for cancer. People talked about it for pain. But when we really got into using it in the context of what we do, which is detoxifying people, purifying, cleaning them out, the hitches that we would often find when we're trying to detoxify people are neurological hitches, specifically neuroinflammatory reactions. Anyone with multiple chemical disorders, anybody who's had Lyme disease or mold toxicity will know that they try some new therapy and the first thing they get is brain on fire. They get neuroinflammatory problems. And it can be brain fog, it can be anxiety, and sometimes they don't even really feel it. But this is a phenomenon that happens mostly around the glutamate receptor, and you get hyperexcitability of the glutamate receptor, and that can cascade throughout the autonomic nervous system and cause dysfunction anywhere in the body. As we got to use CBD in our protocols for detoxification or for treating Lyme or mold, we found that its greatest utility was in settling down the neuroinflammatory reactions that people were having. And when we settled that down, the whole system settled down with it. The autonomics came with it. You'd find people, oh my God, I'm trying to detox them from mercury. They have this chronic kidney stress. So you give them kidney supplements, but it doesn't stop it. You lower the dosage of the detox supplements, and that lowers the pain, but it doesn't detox them. And then we found that it was actually all cascading down from the brain. You had to settle the reactivity in the brain before the body could release and let the healing happen. That is why I say that this is possibly the most important supplement for harmonizing biomedical therapies. We're going to talk about the endocannabinoid system, and the first part of this is just to let us know, hey, the body was meant to deal with cannabinoids. One of the greatest parts of your neurological system that settles a balance between neurotransmitters and immune function is the endocannabinoid system. So we're going to talk about that for a second. Then we're going to talk about phytocannabinoids, the way we can supplement for the endocannabinoid system. Then we're going to talk about the brain on fire aspect, CBD, glutamate excitotoxicity. This is when the glutamate receptors start becoming hyperactive. This is why you have a lot of these children on low glutamate diets or low glutamate stimulating diets. Because it's not necessarily just that they have a massive level of glutamate in their brain, it's that the receptor that's reading that is hyperactive. And that is causing neuroinflammation. Then we're going to talk about what people don't talk about enough, and that's the second part of why CBD works so well, and that is gene transcription modulation. If you ever see me talk about detoxification, I'm talking about upregulating a set of genes for detoxification through a trigger called NRF2. Goes into the nucleus and turns on all the genes for chemoprotection. There's a lot of plant compounds that do it, and one of them is cannabidiol, and some of the other cannabinoids do this as well. So we have something that can calm down the negative effects of these therapies by calming down the central nervous system hyperexcitation, and at the same time can turn up the processes of detoxification, of free radical control, of repair and regeneration. And then we'll talk about a delivery system and how we deliver cannabidiol and why it has such a high bioavailability, and we'll look at it compared to some pharmaceutical bioavailabilities. All right, so just an idea of the endocannabinoid system. It might be easy to get into this frame of mind where you think that the brain evolved somehow with cannabis plants and they have some symbiosis together. But the cannabinoid receptors actually evolved long before the cannabis plant did. In fact, the endocannabinoid receptors, there's one called CB1 and CB2, existed in all primates. In fact, as far as 600 million years ago, we had organisms with cannabinoid receptors. So everything except insects and nematodes and hydra, anything that's a vertebrate has cannabinoid receptors. So they've been there for a long time, and that means that thus we have cannabinoids that we make inside of us. So 600 million years ago, we had an endocannabinoid system, and the cannabis plant evolved approximately 25 million years ago. So this is a plant that makes chemicals that work with our endocannabinoid system. The endocannabinoid system will look at the receptors and what they mediate, but they mediate a lot of neurological function, a lot of central nervous system function, and a lot of peripheral immune function. So the classical endogenous cannabinoids, the two major ones, one is called anandamide, or the bliss molecule, and this is a little bit more of a central nervous system one. And then there's one called 2-arachidonyl glycerol, and this one is a little bit more towards the CB2 or peripheral receptors. But you see those two molecules there, they're very similar. And these are ones that you make in your brain to have controls over neurologic function and its interaction with immune function. So endogenous means it's made by the body, and these cannabinoids have two receptors that they dominantly work on, the CB1 receptor and the CB2. The CB1 is a central nervous system receptor, and the CB2 is predominantly peripherally in the immune system and the organs, but it's more of an immune regulating one. So in the brain, in the basal ganglia, you've got movement effects related to the CB1 receptor. You've got it through the cerebral cortex, the hypothalamus, the hippocampus, the spinal cord. So most of the functions of the brain do have some regulation from this endocannabinoid system. Now, the peripheral CB2 is even vaster in all the different effects that it has. So each one of the organs has CB2 receptors, and the relationship of the CB2 receptors to the cannabinoids controls multiple different functions within the body. So I don't want to focus too much on those. I'd like to spend more time on the brain. But just to follow up on the endocannabinoids, anandamide, first discovered in '92 by the iconic now Professor Meshulam. Ananda is Sanskrit word for bliss. It's something that's found in chocolate. It's something that's produced during meditation, and it's thought to be responsible for runner's high. So the CB1 receptor, the one in the brain, is responsible for more of the psychotropic effects of cannabinoids. So THC dominantly hits the CB1 receptor. The CB2 receptors are more related to control of cytokines and inflammatory responses, modulating the immune system. 2-arachidonyl glycerol is synthesized from omega-6 and is more of a CB2 type receptor. They found it in animal milk and human milk. And we don't have to spend much more time on that. But they all have receptors at the synapses, and they're involved in the dance of how all of your neurons communicate together. So now getting to the ones that were active in supplementing, the phytocannabinoids. So we all know THC, very strongly psychoactive. It's a strong agonist of the CB1 and CB2 receptor, but it first hits the CB1. So its most dominant focus is on the CB1 receptor. Now later when we talk about gene transcription, you'll see that THC has very little effect on gene transcription, meaning expressing your different genes. So phytogenomics is the study of plants regulating how you express genes, and it's a huge part of how herbal medicines work for us. But THC doesn't really do that. THC is more acting on these different receptors. Now, CBD is known to be non-psychoactive. Now, that it doesn't mean that it doesn't have a neurological effect, because it strongly modulates the receptors themselves. And so in the case of THC, CBD actually modulates which of those receptors the THC hits. So THC dominantly goes after the CB1 receptor when it's alone, and that gets you high. It has a very psychotropic effect. But when you add CB2 in, it nudges the THC over to the CB2 receptor, which gives the THC then more of an immune-enhancing effect, an immunomodulatory effect. You get, and then you also get more of the anti-inflammatory effects of it. So when you're doing cannabis medicine and you are using THC, it's crucial to include CBD in it. But CBD on its own, contrary to a lot of belief that you need to have THC involved, CBD has very strong effects all by itself. And a lot of that, and back to the feeling of changes in mood and changes in well-being in CBD, from taking CBD, is because of the way it stabilizes the glutamate receptor, takes away glutamate excess, and produces a more GABA-toned neurological system, which makes you feel more relaxed. But now another thing that CBD does, and I forgot to put the slide in here for it, is inhibit an enzyme called FAAH, F-A-A-H, fatty acid amylase, or fatty acid amide hydroxylase. And this enzyme breaks down anandamide, and CBD represses it. So when you take CBD, you keep higher tones of anandamide, which give you more of that bliss feeling, which is why you'll take CBD and you'll feel good from it. Some of that effect is from stabilizing neuroinflammation and taking away disruption in your neurological field, and the other side of it is from some weak effect right on the CB1 and CB2, but that augmentation of anandamide. So CBD, as I mentioned, weak agonist for CB1 and CB2. They're now finding that it does bind to CB1 a little bit, but not very much compared to, say, THC. It does modulate anandamide very strongly. It modulates the way that THC hit those different receptors. And it's also very strong anti-inflammatory, which it's very well known for, and the transcriptional modulation I mentioned. So really what I want to talk a lot about now is brain on fire. All right. So in autism, neuroinflammatory action is one of the biggest things that's going on. And as you know, I'm used to treating people with mercury toxicity, mold toxicity, Lyme disease. Neuroinflammation is a massive part of what's going on there. A lot of this is mediated by glutamate excitotoxicity. Glutamate excitotoxicity is making more of a name for it now because of traumatic brain injury. In traumatic brain injury, you have glutamate receptors being hyperactive, and it actually leads to neuron death. So glutamate excitotoxicity actually destroys neurons. So let's look. This is our artist rendition of glutamate excitotoxicity. I think that was from a Brian Eno album. So now looking at the biologist's view of it, in neuroinflammation that's it started from immune reactions, you have what's called glial activation. And so you have activation of the microglia. Often astrocytes are recruited into the brain through a leaky blood-brain barrier. These immune cells aren't supposed to be into the brain, but they get into the brain under stressful circumstances, and they activate the macrophage from, or the glia, the microglia, from a nice microglia to a mean microglia. Now, the mean microglia is going to release reactive oxygen species like peroxynitrite. It says here neurotoxic agents, but what these are inflammatory cytokines, the strongest of which is probably TNF-alpha, and after that IL-1 1. So these are supposed to create radical inflammatory reactions, which are pro-oxidant reactions in order to kill infections. But here they're happening even in the absence of an infection. And so this is creating inflammatory processes in the brain that are not supposed to be present in the brain. And these are generally thought of as chronic processes, where you have this chronic low-level inflammation always turning over in the brain. So what are the kind of things that activate this? Well, you've got Lyme and co-infections that often go into the brain. Neurolyme. Neurolyme is the craziest form of Lyme, so to speak, and because your body may look fine, but you're not reacting well. Your reactions are way off the cuff. Viruses. So here I have human herpes virus 6. That's the most common neurological virus. And there's a little bit of a chicken or the egg thing, because most people have herpes 6 in their nervous system, but it's not being activated. So something is causing it to be activated, and this is possibly recruiting other astrocytes into the brain and augmenting these inflammatory reactions. Beta amyloid plaques can cause this kind of immune activation. Though recently they're seeing that in Alzheimer's, the presence of beta amyloid plaques does not create Alzheimer's symptoms. It's inflammation underneath or within the context of the amyloid plaques that causes the symptoms. And then they're finding that there are background infections behind all of that. So there's a number of other infectious sources that can get into the brain and cause this inflammation, but also some of the environmental toxins, like mold toxins, can cause not just direct toxicity to the brain, but can cause immune reactions. That's one of the stranger aspects of mold toxins, is that they activate the immune system as well as being direct toxins. So this is a really good I apologize. These projectors are a little blurry, but this is a picture of self-propagating neurotoxicity, where you've got inflammatory triggers that are more immune-based triggers, but they can, now you'll notice over here, lipopolysaccharides, which are parts of bacteria that you get from leaky gut or that you get from chronic infections in your jaw or sinuses. They can be activating the immune system. A number of these different toxins, you have paraquat, rotenone, those can be activating the immune system and causing the glia to release all these pro-inflammatory cytokines, which are then causing damage to the neurons. And the neurons are then turning around and releasing more immune signaling compounds that are amplifying the inflammatory release of the microglia. So this wheel is turning like this. And the wheel can be turned or initiated from the inflammatory trigger that's more immune-based, any toxin that amplifies the NMDA receptor. That's the glutamate receptor. Many toxins, mechanism of action is through stimulating the glutamate receptor. So which ones do this? Mercury, very well known for doing that. Oh, look, everybody's favorite, glyphosate. And guess what we're going to show later that blocks glyphosate toxicity. Mold toxins do this. Lyme toxins do this. EMF stress stimulates the glutamate receptor, and if you hang out at enough of these conferences, you'll know that for sure. Anybody dealing with a little bit of neurological stress here? Product toxins can do this. I mean, how many times do you get into the situation where yourself or the child is having bad neurological symptoms, and you notice they're super gassy at the same time? You give them something to control dysbiosis, some antimicrobials, artemisinin, biocidin, and all of a sudden, this calms down and the brain clears up. They tend to go together. As the GI inflammation goes up, the neuroinflammation goes up. They tag along together. And is that because of the toxins coming through the GI tract into the brain? And, with the GI inflammation comes the leaky gut, and with that comes the endotoxin. Then that inflammatory trigger opens up the blood-brain barrier, and the whole thing goes into this cycle together. Creates the proteins that create the tight junctions. That's why CBD is so good on GI inflammation. But at the same time, it's more known for calming down neuroinflammation. So it's going to help you in both sides. Notice that I listed glyphosate twice here. That must mean that it's double toxic. Can't be that I just spaced out and put it in twice. All right, so we've got our reinforcing wheel of neurotoxicity there. Where are we going to block some of these things? So first paper, cannabidiol in vivo blunts beta amyloid induced neuroinflammation by suppressing IL-1B and INOS expression. All right, so what that's saying is beta amyloid is causing this neuroinflammatory cascade, and CBD is blocking it by blocking the release of the pro-inflammatory cytokines from the astrocytes. It's stabilizing, from the microglia. It's stabilizing them so they don't put in the pro-inflammatory cytokines. Next one, decreased glial reactivity, meaning calming down that glia so it doesn't freak out and start sending out pro-inflammatory messages. So decreased glial activity can be involved in the antipsychotic-like effect of cannabidiol. So THC has been shown to increase psychosis because it doesn't have that anti-inflammatory effect. And any of you who have been to Colorado and had some of that high THC material out there will notice that you become slightly psychotic from it. That was a joke. But it's not really a joke. I mean, it's, really high THC, wham, it really hits the brain. If it doesn't have the CBD with it to modulate that effect and bring more of a CB2 reception activity and to stabilize the glutamate receptor. Because these things do create free radicals. So THC alone is known to create psychosis, whereas CBD alone is known to block psychosis. It's antipsychotic. So here, it's blocking the hyperexcitability of the glia, and it's blocking their release of the pro-inflammatory cytokines. Neuroprotective antioxidants and marijuana. So it shows THC, but more dominantly CBD is able to stabilize the glutamate receptors. The glutamate receptor is called the NMDA receptor, and if you read these papers, most of this toxicity is mediated through the NMDA receptor. And now the really interesting part of this, and in fact it was in the previous one on the psychosis, is CBD is not just turning down the glutamate receptor, it is stabilizing it. In this case, they were creating psychosis by creating hypofunction of the NMDA receptor. Usually it's overfunction. And there's plenty of papers on it bringing down an overfunctioning glutamate receptor. And there's also ones on it bringing up an underfunctioning receptor. So it's stabilizing and normalizing to the glutamate system. We easily get stuck in this idea that glutamate is the devil and it's a really terrible thing and we have to stop glutamate. If you stop glutamate, you would be like a jellyfish on the floor. You would never want to get up, you would never do anything, and you'd never remember anything. Glutamate is responsible for all of your learning. It's for all of your memory. Why? Because you got to remember what's going to kill you on this brutal planet. And when that thing turns over too much, you think everything's going to kill you. She's going to kill me. She's going to kill me. Everybody wants to kill me, and you freak out and you get anxiety. Then we got cannabidiol, and it's okay. And how is it okay? It's okay because it calms down that hyperexcitability of the glutamate receptor. But it brings it to the middle, and that's my friend Joe Cohen, who's a big cannabis physician in Boulder, likes to talk about cannabinoids as being the ultimate adaptogen. They bring high things down, well, sometimes, and they bring low things up, and bring everything to the middle. So here's another one. Everybody's going to love this because it's everybody's favorite toxin these days. Mechanisms underlying the neurotoxicity induced by glyphosate in immature rat hippocampus, involvement of glutamate excitotoxicity. So glyphosate is an excitotoxin. It is hitting the NMDA receptor, it's making it hyperactive, and it's creating neuroinflammation in the brain. So CBD can calm this system at this point. So what I keep going back and forth between is we can We can stabilize, CBD sometimes stabilizes neuroinflammation at an immune level by preventing release of pro-inflammatory cytokines, and sometimes it blocks it at a neurotoxic level, preventing aberrant activity of the NMDA receptor. And then as we go into the next part, you know, not to spoil the talk, but we're going to talk about how it actually upregulates detoxification. And, you know, one more, cannabidiol attenuates sensory motor gating disruption and molecular changes induced by chronic antagonism of the NMDA receptors. So here again, they would beat up the NMDA receptor, and it just didn't know what to do. It would go too fast, too slow, and it was all over the place. And they would do this chemically, and then CBD would stop that aberrant behavior, going hypo and hyper functioning back and forth, and it would stabilize it, bring it back to the middle. Because we need glutamate, we need a properly functioning glutamate receptor. You know, we all know we have these neurotransmitters up there, and they sort of, there's a symphony of neurotransmitters controlling mood and perception and all of our central nervous system functions. But 80% of that whole symphony is a balance between GABA and glutamate. Now, unfortunately, most of the time it's out of control glutamate, which is causing our problems these days. But that's a big part of the brain, is this balance between those two. All right, so cannabinoids and gene transcription modulation. So if you ever see me speak before, I speak all the time about NRF2. Gary's a friend of NRF2, aren't you, Gary? Woohoo! So what's NRF2? We're not going to spend too much time on it, but in this picture here, this is a schematic of the cell, and the circle's the nucleus. And outside the nucleus is a pair of proteins, the KEAP1 and the NRF2. And certain enzyme inducers will come in and tag the KEAP1, and it'll release the NRF2, and it'll go into the nucleus, and it'll stimulate transcription of all of the chemoprotective genes. Now, what are chemoprotective genes? Well, some of them are antioxidant enzymes that are for free radical control. Some of them are phase 2 and phase 3 detoxifying genes that will, or detoxifying proteins, which will link a toxin like mercury onto a chemical like glutathione and then transport it out of the body. Some of them are called molecular chaperones, some of them are repair molecules, but there's this master switch that brings all that detoxification, free radical control, and repair all in one shot, and that's called NRF2. And some of the things that would trigger NRF2 are certain sort of superfoods that we know, polyphenolic antioxidants like green tea extract would do it, and thus they used to call it the antioxidant response element. They thought it was responding to antioxidants. And then they found a lot of sulfur-based compounds from broccoli. Garlic would do it, things like sulforaphane from broccoli. And then they found lipoic acid was very good. That's the one that we use the most of. And then they found that actually pro-oxidants do it. And so if you're looking at a pro-oxidant like ozone or peroxide or that chlorine molecule in ASEA, those are all stimulating it. Why? Because NRF2 is your stress response molecule. You have these light stresses, and you turn that up, and you clear those out. So this is something we like to turn up, because when we turn it up, we clear a lot of junk. A lot of the junk sits around us, and we get immune to it. I saw there was a great term the other day for nose blindness. You know, your teenage kid that sits in his room full of his own stink and doesn't notice it at all. That's called nose blindness. So, you know, our cells get nose blind at some points, and you need to stimulate detoxification and clear away a lot of the junk that's been stuck in there.